This is the BioCentury Show.
The BioCentury Show is supported by the BioCentury Conference Series. Join BioCentury on the road to network, partner, and debate with peers across the biopharma innovation ecosystem. Our upcoming events span the globe and feature topics from translation to company building to deal making and investment. Visit biocenturyontheroad.com for more information.
Hello and welcome to the BioCentury Show. I am Simone Fishburne, Editor-in-Chief at BioCentury. I am really happy to be joined today by David Altshuler, Executive Vice President and CSO at Vertex Pharmaceuticals. He is a lot more than that, as you'll understand in a minute when I walk through a brief part of his resume, let's call it. David joined Vertex in 2015 and leads internal research and external innovation, corporate data strategy, technology, and data sciences. But even before he led Vertex's highly successful research strategy, he had a distinguished career in biomedical science. David was a founding core member of the Broad Institute, a professor of genetics and medicine at Harvard Medical School, a physician at Mass Gen, and continues to teach students as a senior lecturer in genetics and medicine. He is also chair of the SAB at the Wellcome Sanger Institute, among others, and on the board of directors at the Boston Museum of Science. Maybe that we'll get into later. Maybe that is how you understand how to position those things beautifully on the shelf behind you.
Yes.
And these, of course, are only a selection of the activities and accomplishments because we've only got a half hour, right?
More than enough. Let's get started.
Yeah, let's get started. So I'm going to start by tapping into your expertise that we've talked about across that ecosystem and talk about some of the trends and issues right now. Later, we'll dig into your pipeline programs. So I'm going to start off with what I believe is a particular interest of yours, and that is the topic of causal biology. But this has also been a term that's become bandied around a lot. Early in my career, it was translational research. Those are the words you needed to unlock funding and interest from people. And I'm sort of wondering if that's where causal biology has become as a term. So tell me how you think about it at Vertex and how you see this current surge of interest in the phrase causal biology in biotech.
Absolutely. First of all, thanks for having me. And it's a pleasure to be here. I'm going to start with just what it is that our R&D strategy aims to do, which involves causal human biology, and then I'll answer the question more broadly, as you put it. Years ago, studying the industry and many people looking at it noted that the biggest challenge in R&D is just the high rates of failure. That it's estimates are something like 5% of new medicines that enter the clinic actually end up becoming approved medicines. And of those, actually, most are Me Too drugs. So clearly, despite all the talk about we publish papers in academia like I did, and we start, people work in companies, to actually bring a new type of medicine to patients is a rare event. And I think that what became clear to me 10 or 15 years ago as a human geneticist and doctor was that of the medicines that worked, many of them were tied to causal human biology, the actual cause. And of those that failed, many of them lacked causal human biology. I learned that most from actually consulting with and collaborating with pharmaceutical companies that were working on projects that were based on mice or cells in a dish or a model of some kind. So when I had the opportunity to join Vertex as CSO, Jeff Leiden and I, who was the CEO then, is now our chair, said we want to develop a strategy for serial innovation, not just discover one medicine and market it, but a second, and not just one disease and a third, second and third. And in order to do that, we need higher rates of success in our R&D. And one part of that we felt was if you only work on the causes of human disease, as documented in people, you're more likely to succeed. You're not going to succeed 100% of the time. And the other piece of it, and then I'll stop, we can talk more about causal human biology generally, is that for many of those cases where we understand the cause of the disease, but it's not yet a treatment, that's because the technology to solve it isn't obvious. And so whereas most companies, let's say, are perhaps platform companies, they say we have a technology, we're looking for a disease, or they're therapeutic area companies that say, we have salespeople in some area and we want to buy a new drug. We said, let's find those unusual examples of a severe disease like CF or sickle cell or pain type 1, I could go on, where the underlying biology is well understood, but no one's yet solved the problem, and then use whatever technology is needed to try and bring a transformative medicine to those patients.
So I'm actually going to dig into what you talk about, the success rates or low success
Yes.
rates, and specifically, Marty Makary, the nominee for FDA commissioner, has said, why does it take over 10 years to make a drug? Actually, I think every commissioner has asked this question. And I really want to ask you whether we have gotten better, whether we as an industry, I think we could call this a golden age in biotech if we want to, in terms of the technology, the biology, but if we, and ideas like causal biology, but are we actually getting better? Are we getting more productive at making drugs?
Yeah, I mean, I think we are. I would say if you look back, when I look back over my career, I entered medical school in 1986, which it doesn't seem like that long ago, but it's a long time ago. And I think about what's been transformed since then, and even more recently, it's remarkable. The number of diseases for which there was nothing, and now there's truly transformative medicines, and we could talk about what those are. I think they do benefit from the understanding from human genetics and human biology, of course, modeled often in the laboratory in different models. And these new technologies, it used to be just small molecules, then it was small molecules and proteins, but now we have a whole range of technologies to try. And the key thing is to figure out which disease and target goes with which technology.
So, okay, so we can treat more diseases and we have more tools, but I don't know whether that really means we're getting faster or it's costing less, or we have more precision, more shots on goal.
Yeah. So I will tell you a bit about, again, I mostly focus on what we do because this is the only company I've ever worked in. So, but nonetheless, we, in the last 10 years, since we made that strategy in 2015, have put 8 programs into the clinic with first-in-class approaches. The CF, it was the next-gen correctors. In sickle cell disease, those are small molecule correctors. In sickle cell disease, it was using CRISPR. In pain, NaV1.8 inhibitors. In sickle cell, in a type 1 diabetes, stem cell-derived islets. For 2 genetic kidney diseases or genetic kidney disease molecules, and 1 alpha-1 antitrypsin, a lung disease. And of those, we're now approved for 2 drugs in CF, Trikafta and Alyftrek. The first CRISPR medicine, Casgevy, with our colleagues at CRISPR Therapeutics. The first NaV1.8 inhibitor, Genavix, just launched, and we have 4 others in phase 3. Inaxaplin for APOL1-mediated kidney disease, Zemilacel, Doug Melton's stem cell-derived islets, now in pivotal development for type 1 diabetes, povetacicept, which we bought last year. We bought Alpine, and then our NaV1.8 inhibitor in diabetic peripheral neuropathy. And then 1 alpha-1 antitrypsin had mechanism of action translate to humans, but the magnitude of effect was too small. So when I look at the sort of likelihood of success in most people's programs, and I look at what our last decade has done, it does seem like if you focus on causal human biology where no one's ever had the technology, and you bring the right technology, you can have a higher success rate.
So I'm going to come back soon to those specific programs because I think they're very interesting, and I've got some questions around those. But I want to stay for a moment on this sort of process thing or idea. And, you and I talked about AI. Now, that is often positioned as a game changer. Yeah. So I want to ask, 2 questions. One is whether you believe that AI can change the probability of success. And that is not only necessary in drug discovery, but to what degree AI can be a game changer. And the other is whether Vertex is adopting AI at any point of the R&D continuum and how.
Yeah, so first of all, clearly AI is a game changer. There's no doubt about it. And I think that obviously the new tools, generative AI, large language, are all remarkable. As with our view of R&D, it's all about picking the right business need, or in the case of, in the case of AI, or the right disease, the right target. In the case of AI, that would be what problem, and then the right tool. Do you need AI, or is there something else? We don't believe in platforms, hammers looking for nails, whether it's like a technology for therapeutics or it's a computer technology. The reason for that is that no technology, like if you look at why it takes a long time to discover a medicine, it's not one thing. One thing I've learned, having been now CSO for 10 years, is it's not one thing. It's like 100 things. It's 500 things you have to do right. And AI can help with multiple of them, but it can't help with others. And so if all you think about is the tool, you might accelerate one piece, but you're actually not going to accelerate the endpoint because you haven't solved the other problems. And often I find some of the people doing this don't actually know what the problems are. If you actually say to them, why does it take a long time? What is it that doesn't work? Why is it XYZ? They don't actually know. And they're trying to say, well, we have a tool. I think if you can combine that tool with the right problem, you can make a big difference.
Can you talk about the areas where you've seen it make a difference inside Vertex or outside?
Absolutely. Absolutely. First, I would say, that if you go back in time, one might have heard recently companies saying, we have a new technology. It's going to allow you to make drugs without having to do screening. In fact, you'll just use a computer and you'll have a 3D model and you'll bind the molecule to it and you'll cure lots of diseases. That is the business plan of Vertex Pharmaceuticals in 1989. If many of you have read The $1 Billion Molecule, you could just substitute AI for, X-ray crystallography and computers. It's the same claim. And has it turned out that X-ray crystallography is incredibly important in computers to drug discovery? Absolutely. But was it sufficient to actually, transform it? It was a tool. And way we use the tool, I'll just give you examples from R&D as well as the business, just 3 quick ones. In our pain program, our CF program, we know a lot about the proteins and how they work. We have cryo-EM. We have thousands and thousands of molecules with structure-activity relationships. And yes, we use AI to try and say, what can we learn from those and how can we design new molecules? But if we didn't have all that expertise, and if our people didn't have the right assays and knowledge of where to use it, I don't think it would be that helpful. In development, we have lots of programs like where we've never done a trial before. And in the case of, let's say, APOL1, no one's done a trial before.
Right.
We use AI to try and find the patients and...
That's what I wondered, because people don't talk a lot about that. But
Yes.
I'm wondering whether it can really change the clinical trial paradigm,
Absolutely.
not only in common diseases for endpoints, but also
Right.
in patient recruitment and selection.
Exactly. So it's in the discovery part, yes, it's useful. In actually preclinical development, it can be very useful in terms of, for example, going through huge amounts of data that normally you have a person. Clinical trials, it's both useful in terms of trying to find the patients. But I'll give you another example that Vertex has been working on is there's a lot of paperwork you need to fill out for trials. One example is that when you have, a protocol for your trial, and then you have, let's say, 200 sites, there's a lot of forms you have to fill out with the protocol. And up until recently, we would have human beings do it, and then human beings would check it. Now, much of it's done, still a lot of it's done by human beings, and it's all been tested with A/B testing and blinded statistical analysis. But we found there's a substantial part of that an AI model can do well. It doesn't replace the human being. It's not going from, 8 hours to 5 minutes, but it makes those individuals more efficient. Yeah. And there's lots of examples like that also in commercial manufacturing. We're using it all across the company. We're just not using it generally and expecting someone to figure it out. We're looking for the business need and applying it where we see the value.
And that makes a lot of sense, because I personally feel there's just like too much emphasis on AI and discovery, whereas actually it's probably going to be most leveraged, most fruitfully earlier, later. And even as you say in the, putting together documents and so on.
Exactly.
Go ahead.
Oh, I just say one other thing, when people say, and I've heard people say that AI is going to transform drug discovery, let's say from 15 years to 3 years. If you just look at how long, and then they talk about binding to a protein, you go, well, that part actually doesn't take very long. Like that part actually is pretty quick. There's actually, and you have to actually make the molecule, show it's safe, PK, then you got to phase 1, phase 2, phase 3, you got to do regulatory filings, you got to commercialize it. And I just don't think that if you know what those things are, you can find applications. I will say that, you asked about speed, and Vertex has launched 8 drugs since 2011. Telaprevir, Kalydeco, Orkambi, Symdeko, Trikafta, Casgevy, Aliftrek, Jernavix, all either discovered in-house or in the case of Casgevy with our partner CRISPR Therapeutics. And our average time from let's say, patent filing to approval is about 7 years. And if you look at that Nature Biotechnology paper from 2016 or 17, it says 12.2 years. And if you ask why we're faster, some of it is, things like we talked about, but a lot of it is actually not doing shots on goal. If you do shots on goal, meaning that you do hundreds of things and then you wait for the data to do the next step, and then you wait for the data to do the next step. And if you know that there's years of work to manufacture or do the IND enabling studies or the chronic tox studies, you realize that many of the years are actually committee meetings. It's like that. Whereas what we do is we pick a small number of projects and we invest at risk in a principled way, not excessively, to try and say, if it's positive, how can we be ready for the next step? And we've cut years off the process just by prioritizing high quality projects and then eliminating white space.
Right. And so, I think going back to what you said before, it's possible that AI might actually reduce the costs and increase the probability of success. I do want to also just note in fairness that several of your products are in the same indication. So, you've built the commercial force and you've done a lot of the learning there. And so you're building on that with each one.
Oh, no doubt. No doubt.
Yeah, I'm going to take us to a short break because, we're going to run on and on and on otherwise. And when we come back, we'll pick up the conversation.
Sounds great. Thank you.
The BioCentury Show is supported by the BioCentury Conference Series. For 3 decades, BioCentury has helped biopharma executives and investors make business-critical decisions and build larger networks with peers across the biopharma innovation ecosystem. Our goals are simple: gather the right people in the room to partner, network, and debate critical issues facing the biotech industry. BioCentury's upcoming events span the globe and feature topics from translation to company building to deal making and investment. We hope to see you on the road at an upcoming BioCentury event. Visit biocenturyontheroad.com for more information and to register.
I am back at the BioCentury Show with David Altshuler of Vertex, in the middle of a really exciting and interesting conversation. So, David, you talked about in the first half, you mentioned that Vertex is the only company you worked for, or biotech company anyway,
Yes.
and you really have had one foot in industry and one foot in academia. And I want to spend just a minute or two on something that's long been a passion of mine, which is the academia industry interface.
Yes.
As you know, we have developed conferences, grand rounds around this. Now, I know there are inefficiencies at this interface, operational ones. I don't want to talk about that for now. I want to ask you a scientific question.
Yes.
Whether you think that there are areas of translational science where advances in academia could really create step changes for specific areas of industry. Have you got thoughts about that?
Absolutely. And I think that, looking forward is always hard to predict the future. But, if you look at, I'll just talk about our pipeline for a minute, CRISPR, when I was at the Broad, I was the chief academic officer. We hired Feng Zhang, and I had the benefit of sharing a floor with him at the Broad. And I had nothing to do with it, but he, had his work on CRISPR. And that work, and then BCL11A was discovered, the target we did, by Stu Orkin, Vijay Sankaran, and then worked up by them and Dan Bauer to understand the mechanism. So the CRISPR program we did would not have been possible without the work of Stu Orkin and colleagues to discover and understand BCL11A, and the work of Jennifer Doudna, Emmanuel Charpentier, Fung, and others to develop CRISPR. I think similarly, I won't go through in detail, our work in type 1, Doug Melton worked on that in academia for 25 years. I took my first class in grad school from Doug in 1988, 37 years ago, and he was chair of my thesis committee. And I remember when Sam, his son, was diagnosed with type 1. And, at that time, Doug worked on frogs, and he was the leading biologist, one of the leading developmental biologists in the world. He spent 25 years trying to develop these islets. His daughter also developed type 1. And now we're in pivotal development, but that would never have happened, not only without the work Doug did, but there's so many pieces of that puzzle that were advanced in academia, not only to differentiate the cells, but to measure things, to grow things. So I think that the interface is incredibly important. I think without, funding of basic science in universities, the sort of America's leadership in biotechnology will certainly slow. But I think that the key is to have interactions between them. And I think that while there is a existing and necessary and valuable approach through VCs, sort of academia, found biotechs, work with companies, one of the things I believe is really important is just teaching and interacting. So, whether it's, when I teach, by the way, if I go in, I give a lecture about genetics or something like that, just the things I used to be a tenured professor of. But I do think that if students meet people in industry, and I also created a fellows program where people could come for 1 year after their MD or PhD and work on a project here and then decide industry or academia, that if we interact more, then actually the academic people see, if they want to, is there a problem that might be helpful or interesting to solve? And then the people in industry will actually have trusting relationships with people so they can collaborate. I think that that is a very valuable thing rather than throwing things over a wall.
So, David, I have more questions, but I can't go on now without referring to what is behind you, some of the interesting things on your shelves that you're showing us. I know the Flutie Flakes is something of particular interest. So
Yes.
walk us through one or two of those items. And those who are only listening and not watching, this should be your impetus to get on YouTube and actually look at the video.
So I did not put this up for this interview. This is
No,
like,
I will guarantee that you didn't.
decades of work.
Correct.
But what I would
Yeah.
say is, that's my wife right there and those are my parents right there. That's actually where I went to high school. My kids, actually, I feel like I should change it because my kids are all over the place. I'm sorry, Zach and Jason, you are here. And then Flutie Flakes, I've lived within 5 miles of where I live now since I was 2 years old, and I'm 60. And that's like 8 blocks from BC was where I grew up. And Doug Flutie is a couple of years older than me, and I was obsessed with him when he was a high school student in 1984 when he went to the Cotton Bowl. And so when, like 10 years ago, I went into an antique store and saw this box of Flutie Flakes, I said, I must own it. And then I keep it just for fun. And the last thing I'll say, you said too, is that document right there. When Felicia Paliuca and Bastiano San and I showed Doug Melton the first result of VX880, the first patient, it was a very moving moment for all of us. And he wrote me a very nice note afterwards. And I keep it there just because, it's very important to remember why we do what we do. And it's unusual to have someone like Doug, who's both a incredibly leading scientist, wonderful person, and mentor to everyone, but also a parent who really cares about the outcome. And so make sure when you get up every day, you feel you know why you're doing it.
So, David, I'm going to stay there for a moment and let's talk about the science side of this, about your diabetes cell therapy programs. So you've got Vertex has one program further in development that requires immunosuppression, a second that doesn't, and the preclinical program with hypoimmune cells, that's in a partnership with CRISPR, presumably using gene editing. And that would be a massive step forward if it was successful. Can you talk a little bit about the biology of these and how Vertex is going about it?
Yeah, absolutely. So, I should state before I do that I trained as a diabetes doctor, and I was for a decade or so was a diabetes doctor at Mass General. And taking care of type 1 diabetes patients, you realize how devastating this disease is. Even if you're able to control your blood sugar and your health, you still have to spend every hour of every day thinking about the fact you have type 1. You have to manage your blood sugar. You have to be concerned about eating and exercise, and the concern that your blood sugar will get too low. And amazing statistic is today, despite pumps and continuous glucose monitoring, only about a quarter of people with type 1 on those devices actually are below the hemoglobin A1C ADA guideline. So there's a great unmet need. And the key thing is to actually be able to create cells, human islets that can restore function. We know the cause of type 1 obviously is autoimmune destruction of the islets. And we knew that would work if you could make the cells, because 25 years ago, it started to be the case that people showed with cadaveric islets that you could cure type 1. So when we started this, or Doug started and we bought Sema, we knew it would work if you could do it. The question is, could you make the cells? We're now in pivotal development. We've reported data. The last data update was on 12 people, and 9 of them were off insulin I won't go through all the data. It's all publicly available. But let me just say, I don't have time, but, the data is very promising. And we're now in pivotal development with an outcome looking for people off insulin with control of their blood sugar, as well as a lack of hypoglycemic episodes. So that's the foundation. And one key thing is to be able to scale that so you can make enough cells. There's 3 million people with type 1 just in the US and Europe. So to go from a handful of people we aim to treat in the long run, 1 million people if they want it, that is a huge task. The second is, how do you manage the immune system? And we're using standard immunosuppression in VX880, what's called Zymylacel now, because that was an existing method that worked for cadaveric islets, and we wanted to, show that just the cells alone could replace the cadaveric islets. We have the device VX264, which is in the clinic. We have the hypoimmune editing, as you said, and there's also worth saying there are now, many immune modulatory agents that are developed by biotech companies and pharma companies that have the potential to offer better immune modulation than traditional immunosuppression. So the way we see it is the cells are the foundation. For any of these approaches, the key thing is, can you make the cells and do they work and are they safe and all that? And then there are multiple, if you will, shots on goal as to how to go from what is already, I think, for the right patients, a very favorable benefit risk profile with traditional immunosuppression to, other methods, whether it's a device, gene editing, or another type of immunosuppression that will make it hopefully a potential for even more patients.
So let's go from there to another gene editing program, a very early stage, of course, which is in DMD.
Yes.
And you want to restore then, full-length dystrophin expression. Question that's still not clear to me is whether we as a field know what level of dystrophin expression is needed for functionality. That seems to be still unclear to me. And there are many different mutations that lead to DMD. So how are you tackling that from a product perspective? What's the product profile there?
Well, that's a great question. And I think, DMD is, the first gene cloned using positional cloning actually was DMD by Lou Kunkel in 1986. And, it's a terrible disease, obviously. And there's sort of two approaches for genetic therapy. One is, which is gene alternate splicing, where you can't fix the individual mutation, but you can fix skipping over an exon, you can restore normal function. And the gene editing approach that we've been pursuing, we bought a company called Exonics in 2019. Eric Olson had founded that company. And the idea is to do that with gene editing rather than an oligo, so it would be a one-time therapy. And the data in models is extremely promising, and I think we do know how much dystrophin you need based on Becker's muscular dystrophy, other human data. The challenge has been delivery. We originally were planning to go into the clinic, but our view became, based on our own preclinical data and watching the field more generally, that AAV was not something we wanted to pursue. So we're working on non-viral and other types of delivery. With regard to, on the other hand, one thing you said was about microdystrophin. You might say, why are we working on dystrophin gene editing, which is obviously you have to do different exons. It's more challenging when people are doing microdystrophin. And it was a decision we made back in 2019 that we didn't feel there was no human biology to say microdystrophin would work, because there's no patients actually with these 3 kilobase microdystrophins. The smallest human thing is 6 kb. And the model data wasn't so clear. And I would say, based on all the clinical data, it's certainly uncertain what the relationship is between the expression of microdystrophin and clinical benefit. So we continue to focus on trying to generate full-length or near full-length dystrophin, even though it's taking longer, because we actually believe there's greater benefit that can be had than is being seen today.
And so you'll have multiple edits inside the...
We do one at a... The goal would be one at a time, but yes, you would be able to make each one, and hopefully if the first one worked well, you'd be able to quickly do the others. But like I said, I want to be clear, this is a long-term project. We are not in the clinic or near the clinic, although we have the gene editing part down. But I do think that, I hope that the potential for patients with DMD is even greater than there is today, if it's possible to restore highly functional full-length dystrophin at an appropriate level.
So I've focused on these because I think we know less about them, and I think they're incredibly interesting. You, of course, also have a pain program and really interesting programs in renal diseases.
Yes.
But we're almost out of time. So what I want to ask you now is, you've made your name and Vertex's fortune, let's say, in cystic fibrosis. And now you've extended to several other therapeutic areas. So maybe even incorporating the other ones I just mentioned that we didn't go into in detail. How are you, it seems difficult to me to translate the learnings from one to another, or do you have to build a knowledge and talent base in each of these different therapeutic areas at a strategy level, which is your role? How do you go about that?
Yeah. First, let me be clear that the position we are now is the corporate strategy that Jeff Leiden set back in 2012. I was on the board at the time.
Fair point, yes.
And the idea was recognizing that innovation is hard everywhere. And the view was that in our industry, if you can create transformative medicines for serious diseases, be first in class, and then lead the way, you can create tremendous value, even if, there's the challenge of how do you commercialize them. But when people say, well, we've already got the manufacturing, we've got the commercialization, but we don't have a drug, that doesn't work as well. And in particular, if you thought there were lots of drugs to buy or lots of targets to use, you'd say that's a fine strategy. Just the history of the industry is it's not true. That that simply saying I'm going to work in the same therapeutic area, the same platform. So what we said was we're going to focus on making transformative medicines. And as I said, we have multiple approved and multiple others in phase 3. And we chose specialty markets. And again, this is sort of Jeff's idea, because while it's not easy to figure out how to launch, in a specialty market, it's very different than primary care where you need thousands of salespeople and all these things. And so, yes, the company now, with its success in R&D and launching these medicines, has been working for years and continues to work. And my colleagues in commercial and in medical affairs and manufacturing all have been working for years to do what you're saying, prepare for these launches. And no doubt that requires innovation and learning. But we believe, first of all, those are great teams. They did learn a lot. And many of them, of course, learned things in addition to at Vertex and other places, but that it's a much better place to be, we believe, to have multiple, not one, but multiple additional potential transformative medicines, whether it's pain, sickle cell, type 1, APOL1-mediated kidney disease, IgAN, et cetera, other pain types, that's a better opportunity to have those medicines and now figure out how to launch them and sell them than it is to not have transformative medicines and have a really good infrastructure for how to get them out there. But it's not easy, no doubt. Everything is hard. We just chose to focus on, could you have a lot of transformative medicines and figure out how to sell them and make them, as opposed to we've got really good manufacturing sales, but we don't have the transformative medicines.
Well, that's a great place to end. We're out of time. I know we could do this, and I hope we will do it again. Thank you so much.
Thank you for having me.
The BioCentury Show is supported by the BioCentury Conference Series. Join BioCentury on the road to network, partner, and debate with peers across the biopharma innovation ecosystem. Our upcoming events span the globe and feature topics from translation to company building to deal-making and investment. Visit biocenturyontheroad.com for more information.
Kendall Square Orchestra provides the music for the BioCentury Show. The group connects science and technology professionals and other members of the greater Boston community to collaborate, innovate, and inspire through music while supporting causes related to healthcare and education.